Welcome to the Global Hemophilia Report, a podcast at the intersection of science and storytelling. Produced by Believe Limited and Bloodstream Media and made possible thanks to our featured advertiser, Sanofi. Folks, I turn 40 this year, on November 30th to be exact, and so does my life with hemophilia. The history of my body and the history of this community's science have grown in parallel for the last 4 decades. And in that time, a tremendous amount has happened. Crisis and discovery, fear and breakthrough, the rise of data, and the rebirth of hope. This episode is both personal and historical. It walks through what my experience of hemophilia has been for the last 40 years, alongside what the broader story of hemophilia was during that same period, filled out and balanced by my colleague and co-host, Dr. Donna DiMichele. Together, we'll trace 4 decades of research, data, and human experience, from the 1980s to today. And we'll ask, what have we learned? What have we measured? And what still can't be captured by data alone? We'll dive in right after this.
Sanofi seeks to break barriers for people with hemophilia through groundbreaking science so they can live beyond the limitations of their condition. Learn more about Sanofi's commitment at redefininghemophilia.com.
Segment 1, the 1980s Crisis and Awakening. When I was born on November 30th, 1985, hemophilia care was at a turning point that no one asked for. Families were just beginning to learn that the very treatments meant to save lives had been contaminated with HIV and hepatitis. For many, data didn't feel like protection. It felt like a list of names that we were losing. For me, this was the era that I was born into. Before I could even walk, science had already changed the course of my life. Dr. DiMichele, for those who weren't there, how did that crisis reshape research and data collection, and what new responsibilities were placed on the scientists and clinicians of that era?
Well, Patrick, as you say, it was a very tragic turn of events. The 1980s were. It was a very, very scary and a sad time in hemophilia. You know, the hemophilia physicians at that time were already worried about transfusion-transmitted infection with the new factor concentrates. It was mostly about hepatitis, hepatitis B, and what we called at that time non-A, non-B, later to be known as C. In the meantime, in the late 70s and early 80s, this immunodeficiency syndrome was beginning to appear, of course, in the gay population. And since the CDC was very interested in their primary role was surveillance for infectious disease, they were already in high gear and in surveillance and surveilling for what was going on and the symptoms that they were seeing in this community. Thanks to some of the astute observations of physicians at that time, who were beginning to see changes and some of the same symptoms in the hemophilia population, the CDC became aware of the same sort of panel of symptoms in the hemophilia population. And it didn't take long at that point to make the connection that whatever, and at that time it wasn't immediately known what this virus was, but whatever it was, it didn't take long for people to recognize that it contaminated the blood supply. And because it contaminated the blood supply and the clotting factor concentrates at that time were all plasma derived, it had contaminated big time. Given how factor concentrates were made, it really contaminated the clotting factor, both factor 9 and factor 8 concentrates, really badly. The severe hemophilia population was the most affected because they were infusing most. But understanding that this was a bloodborne infection, and the hemophilia patients, unfortunately, being the canary in the coal mine, really changed the whole approach, the whole national approach, the whole international approach to blood safety. Because right about the time you were born, what was called then HTLV3, later to be known as the human immunodeficiency virus 1, was identified. both in France and at the NIH. And so right about the time you were born, testing for this virus became possible, testing individuals and testing the blood supply. And the sad part of this was that, of course, we began to recognize just how much HIV had infected our hemophilia population. But we also began to be able to screen blood, and it began this whole era of not only blood screening, but virus inactivation. You were kind of born right before the blood supply became safe, because we will say, and it is true, the epidemiology shows that as of January 1st, 1986, no patient became infected with HIV because of the ability to test, the blood supply, the ability to inactivate viruses. And there was a lot of pressure placed by the community on the factor industry to indeed treat the factor concentrates to inactivate virus. So there was virus screening, virus inactivation. And, blood safety became the priority across government organizations, the FDA, the NIH, the CDC, and really changed the whole approach to blood safety in this country, and like I say, internationally. And thanks to the ongoing efforts, in the hemophilia community, the government, transfusion services, this has been a priority for this country in terms of surveillance for new viruses in the blood supply and in any of the derivatives of the blood supply has been a very big priority for this country. And that's why sometimes what's going on in the government right now is very scary, because, it has been the safety net that we've come to rely on for the last 40 years. But I think the lesson from that period, the tragic lesson from that period, is that with innovation comes risk. And the innovation that was the factor concentrate was astounding for the hemophilia community. They felt that they had taken a giant leap forward. And the risks that, were so unanticipated, but so devastating, really have hopefully taught us the lesson that any innovation may come with risks, and that we never need to let up on safety surveillance. So I think that was the lesson from that period. And I'm glad you were born when you were, because, your risk was significantly diminished within 2 months of your birth in terms of getting infected. Thank goodness.
I feel tremendously lucky, and that's a very rich history. Thanks for walking through that with us.
Kevin lives with hemophilia A. He enjoys hanging out with friends, cooking, and playing guitar. But what you don't see is how living with hemophilia has impacted his communication style. Kevin bravely shares the hurdles he faced in communicating about his hemophilia with friends, family, and providers, and highlights the importance of transparent, patient-focused conversations that go beyond treating symptoms. Scan the QR code or click the link in the show notes to hear Kevin's full story and learn more about how living with hemophilia can impact communication.
So, fast forwarding to the 1990s now, an era of recombinant factor replacement and inhibitor development. So by the time I was a kid, recombinant factor replacement therapy was beginning to replace plasma-derived products, and this concept of prophylaxis was being spoken about, it seemed, everywhere. The idea that you could prevent bleeds instead of just reacting to them, it changed everything. For a community still carrying the trauma of the 1980s, it was the first real glimpse of safety, control, and possibility. But these new therapies, as they became widespread, another challenge emerged: inhibitor development. The very thing designed to restore normal clotting could, for some of us, trigger an immune response that made treatment far more complicated. For me, as someone who developed an inhibitor, in spite of being in an era of safe and effective recombinant factor therapies, I was forced to rely on plasma-derived bypassing agents, which were not nearly as effective at controlling bleeding. So, Dr. DiMichele, let's talk about this introduction of recombinant factor replacement products. What did that mean for patients and clinicians at the time, and how did it lead to a growing focus on inhibitor development in the early to mid-90s?
Yes. Well, I'll tell you, it was a game changer. and it's said, I mean, it revolutionized hemophilia therapy because the plasma scare and the blood safety issues were still very much top of mind. And although plasma concentrates were considered to be safe, as I said, starting in 1986, the idea of the recombinant clotting factor was amazing because it totally obviated the concern associated with blood safety. And not only was it genetically engineered and therefore not blood derived, but it also was able to be infused in a very small volume, and it made infusion much easier to do, self-infusion much easier to teach, and the ability to treat with more frequency much easier to do because it was short, it was easy, and the burden of concern about blood safety was, like I said, totally removed. Now, I want to say though that there was initially some very healthy skepticism about recombinant because it was genetically engineered and it wasn't considered to be natural. And a lot of that skepticism, I must say, came from the adult hemophilia patients, many of whom had, been unfortunately lived through, and many of them had been victims of the HIV crisis and blood safety issues, and they were wary of innovation because of exactly what they had gone through, and I think rightly so. But as I said, the whole issue of recombinant really changed the way we treated and the way we thought about treatment, and then the focus, as you said, when you talked about prophylaxis, the focus became on the orthopedic, morbidity that was being associated with hemophilia and what we could do about it, because now we didn't have to worry about factor safety. We didn't have to worry about factor volumes. We could even treat very young children very easily with the smaller volumes that were, they were 5 cc volumes of small, and some of them became 2.5 cc volumes. So the smaller volumes that we were able to infuse. And so we, orthopedic disability, even before the inhibitor issue, orthopedic disability was really on the radar. It was the concentration for most hemophilia treatment centers. And, how do we treat, the joint disease, the musculoskeletal disability that was already there? And several things were happening. People were trying to document this in a much more coordinated way, both nationally and internationally. There were persons who were affected who had orthopedic issues were able to undergo surgery a lot more effectively and safely. And so orthopedic surgery became much more feasible and much easier to do. Procedures such as arthroscopic and radioactive synovectomies became much more frequent as a way to try to manage bleeding that was going on in target joints at the time. And then we had only been able to look at joint disease using x-rays. And then the MRI came in, and the MRI, which was able to give us a picture of the soft tissues, of the synovium, of the cartilage, and all the damage that was going on in the joint, even before we saw it in the bones, was really changing the way we were looking at joint disease. Now, in the meantime, our European colleagues were starting to publish on their long-term results with using prophylaxis. There was reports out of Sweden where they used very high dose prophylaxis, and out of the Netherlands where they were using low dose prophylaxis. And the issue of prophylaxis became a very important one, and interestingly, slightly controversial here. Controversial mainly from the financial aspect, because the amount of clotting factor compared to on demand that was being required to effectively cut the bleeding rate, as we were able to measure it then, was up to 6-fold higher than individuals were getting on demand. And even at that time, clotting factor concentrate was not inexpensive. And so, the question of really understanding the benefit of prophylaxis was top of mind in the United States, and the joint outcome study was born, and that was a randomized control trial of prophylaxis versus no prophylaxis in pups. That was conducted by Dr. Marilyn Menko Johnson, and still is the study that confirmed unequivocally the benefit of prophylaxis. And once that study was completed, in the early 2000s, once that study was completed, prophylaxis became the standard of care. But back to inhibitors, it became, one of the things that happened with recombinant licensing studies is that because they were genetically engineered and the proteins were somewhat different, the part of the monitoring for safety was the monitoring for inhibitors. Now, interestingly enough, plasma-derived clotting factors had never really been monitored in the same way in licensure studies. So we didn't have a baseline, but what was beginning to be recognized in these early product licensure studies was that about 30% of the previously untreated patients in these studies were developing antibodies. And so that's how we began to define the inhibitor rate as 30%. And everybody was like, wow, this must mean that, recombinant clotting factors are much more immunogenic. And it certainly both raised the interest in factor VIII, particularly factor VIII. This is primarily relegated to factor VIII. The interest in factor VIII immunogenicity, but also created the new crisis of the time, and that was the development of antibodies, because there was a sense that these antibodies were developing in higher titer, they were developing earlier. There was a lot of debate at that time, which studies had to eventually sort out about whether we were giving higher dose. Was it because we were giving higher doses? Was it because we were treating more frequently? Were individuals getting more clotting factor over a period of time? Was that the issue that was inducing the immunogenicity in factor VIII, or was it a difference in the product itself, the difference in the factor VIII molecule because it was genetically engineered? So the focus then became, okay, how do we begin to understand inhibitors? And of course, how do we begin to treat inhibitors? And so a lot of, you ask about how does that change surveillance? Well, suddenly everybody was interested in natural history of inhibitors. What had we been missing all of this time? When do inhibitors develop? After how many exposure days? When can we anticipate? What are the predisposing risk factors to inhibitors? What are the ways in which we treat that make inhibitors more likely to appear? And there were international studies going on, the PEDNET study in Europe. And thanks to, by the way, CDC's ongoing interest in hemophilia. So once they became interested in hemophilia during the AIDS crisis, they never stopped being interested in the surveillance of hemophilia as a chronic disease. And in 1998, the Universal Data Collection System from the CDC was established in collaboration with the hemophilia treatment centers. And we were beginning to collect data on persons with hemophilia, and that included a lot of joint outcome data. And that also included inhibitor data. So there was a lot more focus on joints and inhibitors during that time. And then, the question was, how do we treat this? And there were reports from Europe about using high doses of clotting factor VIII over long periods of time, often given daily to eradicate inhibitors. Other countries were reporting doing it with less and less frequent schedule. And so the interest then became internationally in how do we collect this data and how do we understand this process called immune tolerance. And I was asked to create a registry within North America to understand immune tolerance practices and outcomes. And it wasn't being used a lot at that time. But with the increase in inhibitors, people started to get more interested in it. And the information that we had gathered from the registry really enabled us to design the parameters of the International Immune Tolerance Study, which then began in the late 1990s and went on into the 2000s to help understand how we can best do, immune tolerance. So all of that was kind of going on in the 1990s, and so and it was to be continued in the 21st century because the story didn't end there. But a lot of people think that, oh wow, there's so much happening in hemophilia now, but the 1990s was that period, 30 years ago.
Rich lives with hemophilia A. He enjoys gardening, home improvement projects, and traveling. But what you don't see is how living with hemophilia has impacted his ability to live an active life. Rich bravely shares his experience living with this rare disease, its impact on travel and activity, and the importance of overcoming hemophilia's impacts to do what you love safely. Scan the QR code or click the link in the show notes to hear Rich's full story and learn more about how living with hemophilia can impact travel and activity.
Well, and to your point, it didn't end there either, so we'll just keep our train rolling into the 2000s, our third segment, where we'll discuss overcoming inhibitors and some global disparities. So fortunately for me, by the age of 12, thanks to immune tolerance induction, I was able to overcome my inhibitor. I underwent synovectomies on my most compromised joints and began to see a quality of life that was much different. By the time I was 18 and off in college, my day-to-day experience wasn't that much different than my peers. At the same time, the opportunity to disconnect from my hemophilia was never greater either. And of course, in the case of my brother, who entered college healthy, on prophylaxis, and without inhibitors, well, his story took a deadly turn due to disconnection. So Dr. DiMichele, tell us about the larger hemophilia world in the 2000s. What were we still reckoning with from the 1990s? And tell us about the moves and shifts in the landscape that really defined those 2000s years.
Yeah. Well, the story that, as you bring it up, is very important because one of the things that happened with recombinant clotting factors is we allowed, boys and young men with hemophilia to become much more independent. As we said, self-infusion was easier, and, the opportunity to go to college, to, be away from parents, to, transition into adulthood, without always being aware of the burden of hemophilia became a possibility. And so that was going on, and that was good. And there were other good things happening. I mean, in essence, the first 15 years of the 2000s was like just refinement. Nothing major was happening, or maybe it was things were beginning to happen, but mostly it was this period where, there was a lot of refinement in care going on. Now, one good thing, and it sort of flew under the radar for the youngest people with hemophilia because they weren't affected, was that curative therapy for hepatitis C emerged. And that was a game changer for adults who had been infected, adults and, younger adults who had been infected with hepatitis C. And of course, later went on, because the eradication of hepatitis C later went on to make a lot of these young men eligible for gene therapy. So that was a very important development. In terms of joint health, this concept of primary prophylaxis had taken hold, and people were beginning to look at studies that were beginning to show that remember, we were aiming for trough levels of over 1%, and people were beginning to show that maybe that wasn't good enough. And we're starting to refine prophylaxis in a way in which trough levels were not allowed to get that low, and we're defining maybe trough levels of about 10% that were maybe required to really get to the zero bleeds that everybody was sort of aiming for. And of course, at that time, we started to get extended half-life clotting factors. And at first, the advances were more on the factor 9 side than on the factor 8 side, but longer acting clotting factors allowed us to maybe achieve higher clotting factor levels, so that with infusing less, folks with hemophilia might still be able to achieve trough levels that were a little higher than 1%, and thereby do a better job of preventing bleeding. And then, of course, the 2000s brought us ultrasound, and point of care ultrasound to begin to detect bleeding potentially at the time of visit. Because, sometimes it wasn't clear. Was it synovitis? Was it bleeding? And we started to get another detection tool. So in addition to the MRI, now we had the ultrasound. So there was a lot going on in understanding and optimizing prophylaxis and early bleed detection, and trying to get to this, zero bleed per year kind of threshold. So that was going on. And, because the UDC was capturing all this joint data, people began to get interested in joint disease in women who had symptomatic hemophilia, and the whole issue that women could have joint complications began to emerge, and the whole interest in women and girls with hemophilia started to be, it took a while, but there was that nascent interest in beginning to study women and girls with hemophilia. And in terms of inhibitors, again, a lot was going on. Mostly, although there was a lot of basic science going on to try to understand the immunology of factor VIII, none of which have so far yielded any major prevention strategies. But nonetheless, there was a lot of work going on there. But there was also a lot of work going on in the epidemiology of inhibitors to understand how they develop, when they develop, and how we might be able to intervene. There was still all this discussion about, is recombinant factor giving us more inhibitors? And then there was the SIPET study that actually showed that, yes, indeed, there was a higher chance of getting an inhibitor with that recombinant factor. People are still disputing that. And, but in any case, we were learning a lot more. And then the immune tolerance study got completed and showed, although we couldn't show dose supremacy or inferiority, one of the things that we're able to demonstrate in the international ITI study was that if you're going to achieve immune tolerance, stopping bleeding, particularly early in the course, is very important. And it may be more important than how you give the clotting factor, what frequency, daily versus non-daily. And that set the stage for what was going to happen next. That is the evolution of emicizumab and the ability to do immune tolerance with emicizumab prophylaxis that then allowed people to, now experiment. And that's what's going on now, right? A much less intensive way to do immune tolerance. And then lastly, then the novel therapies. In the beginning of the 2000s, Kathy High was doing her first experiments with gene therapy in muscles, and it didn't yield. This was for hemophilia B, and it didn't yield very high levels of factor 9. But it really set the stage, since she was using adeno-associated virus, it really set the stage for the next set of gene therapy, trials that became liver targeted. By 2017, there was emicizumab. But, just goes to show you how long innovations take. I mean, the first laboratory studies, or the first ideas about using a bispecific monoclonal antibody to replace factor VIII in the activation of factor X, was actually going on in Japan in like 2005. So all of this was going on. It wasn't reaching the clinic yet. And it wasn't until, 2007, toward the end, close to 2020, when, we were started getting this concept that, novel therapies and, might actually be a game changer for hemophilia. And by the way, that was because gene therapy wasn't a runaway success. And when gene therapy became clear that gene therapy was going to take longer to become the curative therapy we were looking for, everybody said, okay, we need other therapeutics to really optimize the care of hemophilia. And then, the race to novel therapies really began, by the latter half of the 2000s. So all of that was kind of all going on at the same time.
So much activity. It's actually quite refreshing for me anyway to hear you talk it all through. It's like a trip down memory lane. Oh yeah, I remember that. Or, oh, that was going on during that time. So much activity. So let's jump ahead. We've already jumped into the 2010s talking about the introduction of emicizumab, which is commercially in the US known as Hemlibra. But let's just double click on a couple of other items. So by the 2010s, data meant more than lab numbers. It meant patient-reported outcomes, quality of life scores, and wearable logs. We started asking, is a zero annualized bleed rate, or ABR, the only definition of success? Or does success mean the freedom to live without thinking about hemophilia every day? And we also, as has been mentioned, cannot overlook the introduction of the first non-factor replacement therapy, emicizumab, a game changer offering people with inhibitors and later those without a completely different way to manage bleeding. For the first time, treatment wasn't about replacing what was missing, it was about reimagining what was possible. And personally, I'll share that for me, this decade of my life is when my storytelling work took off. Documentaries, podcasts like this one, and live events, all with strong advocacy and educational cornerstones. So I watched in my little neck of the woods as science and storytelling finally started to converge in meaningful ways. Dr. DiMichele, I want to ask you specifically now about evidence. What did the shift toward real-world evidence reveal about our prior assumptions, and how did it influence decisions at the Food and Drug Administration or the European equivalent, the EMA, and within pharma research? And if you want to share anything more about the introduction of Hemlibra and how it changed the landscape, I think that would be appropriate.
Once again, when you sort of think back and, when you think back on it all, retrospectively, you realize just how much was going on. But yes, I mean, just like I said, recombinant was a game changer. I mean, Hemlibra, emicizumab, was an amazing game changer. I mean, the studies coming out of Japan, even before the Haven studies were begun, were generating a tremendous excitement in terms of the ability of this bispecific monoclonal antibody that was called emicizumab, how well it could actually prophylax against primarily musculoskeletal bleeding in individuals with severe hemophilia and inhibitors, and then it went into phase 3 clinical trials, and the first Haven study, which again was coming out in about 2017, and the fact that it could be so effective and have minimal, what was felt to be at that time, minimal, and still, I think, minimal side effects, was just a game changer. And it was a game changer not only, as you know, for individuals with hemophilia and inhibitors, but individuals with hemophilia A who didn't have inhibitors, and almost became the standard of care in a very few years. I mean, the adoption of emicizumab over standard clotting factor therapy, even as, by the way, longer, especially in for hemophilia A, longer, much more extended half-life factor VIII concentrates were being developed, like Altuviiia, even with the appearance of these better and longer acting factor VIII concentrates, emicizumab just took off. And it really ushered in this era of novel therapies, and everybody was looking at how the coagulation system could be manipulated outside of the standard, requirements for factor VIII and factor IX in order to create hemostasis. And so, that's when, the TFPI inhibitors and fitusiran and all of these agents that are now just beginning to be licensed, all of that work was ushered in, by the first one, and that was, the emicizumab. Now, you asked me, and by the way, gene therapy was still moving along, but it was undergoing one of its first sort of course corrections in terms of how best to do these studies and how to optimize the vector and how to optimize the transgene. And a lot of that was going on all at the same time. But it was very exciting because all you heard about were novel approaches to treating hemophilia, and you asked, how did that affect the regulatory side of things? I don't know. I think there was, in looking at it retrospectively, I think there was the appropriate amount of urgency to license products that were significantly needed, especially, ultimately for the hemophilia B inhibitor population, but also caution. And I think there was, the right amount of caution and movement, to license these products that were needed. But I think one of the things that became very clear is that the outcome measure of the ABR, and the zero ABR, the, absolute bleeding rate or the average bleeding rate, was actually becoming a moot point. And in other words, standard prophylaxis was so good. And, that when these novel therapies were being compared, they had to be compared against on demand in order to show an effect. And so what began to be appreciated, I think, was the ABR as a blunt instrument, and not at all precise enough to be able to, for instance, do post-licensure studies and show differences among products, to really compare new products to standard prophylaxis. The outcome measures were just not precise enough, and I think people are still discussing that. And the need for more sensitive indicators is really critical, and that's because we have so many choices. How are individuals going to make those choices? What's best for them? How are they going to compare one product against another, for their particular situation? So all of that, I think, is incredibly important. So, of course, data collection has become even more important, right? And that data collection, because, by the way, all of these products are so new. They're like all these potential innovative Trojan horses that are carrying risks, and risks that are unexpected, unanticipated, risks we can't even begin to think about. So data collection has become really, really critical. And it's never been more critical than it is now, particularly with respect to safety monitoring. And but also with respect to therapeutic effectiveness monitoring and developing new clinical outpoints. All of this really needs to go on. And I think what happens is when innovation is happening so fast, data collection lags behind. And I think that now that hopefully we may be stabilizing, this is an important time to, gather our thoughts and say, what are the important questions? What do we need to answer? And how do these questions need to be answered? And there are various ways. I mean, there are intentional studies. I mean, I happen to think the FDA should be demanding more post-licensure studies on all of these products from the companies as they used to. These are called phase 4 studies. I think there are, observational studies. Randomized studies are a little hard to do, but data collection over long periods of time through registries and intentional observational studies is a very important way to collect the real-time data and that we absolutely need in understanding how these products work in a larger population, a more diverse population, and are they as safe and effective as pre-licensure studies are indicating that they are. And I'm glad to see that some of that is going on. The Europeans have had a database to collect safety data on all therapeutic products. It's called EUHAS. And Athens 7 in this country was modeled on EUHAS, and now it's transformed into Athens Transcends. There are several national databases in Europe and in the United Kingdom. The UK HCDO has had long-standing data collection and is currently collecting very good data on safety and efficacy. And, ultimately, this is going to be needed for gene therapy too. And I know that the World Federation of Hemophilia is leading an international effort to continue to collect safety data. So I think all these efforts are good. And I think they're needed, and I hope that we have learned our lesson from the past, and saying that we just can't take our eye off the ball because we never know what's going to go wrong, and so for the sake of our patients, we need to be very, very clear in our intent to survey for safety and efficacy.
Hear to that. Well, that brings us to the 2020s, an era of digital health, gene therapy, and that poses the question, at least for me, what will the next 40 years look like? Today, yes, data does live everywhere. It's on our wrists, it's in our phones, it's in the cloud. Soon, AI will be predicting bleeds before they even happen. At least it feels like we're headed in that direction. And yet, for all this technology, the core question hasn't changed. How do we make sure every person with a bleeding disorder is counted, seen, and supported? Turning 40 makes you reflective. It also makes you a little impatient, or at least it has me, a little bit. We've come so far, but the work of collecting, connecting, and caring is not finished. So what happens next, Dr. DiMichele, and how do we ensure that as data helps inform our practices, our research, our work in the clinics, our work as individual patients taking care of ourselves, how do we ensure that the benefits of all of this are reaching everyone with hemophilia everywhere? And as we have things like gene therapy and non-factor replacement therapies, how do we ensure equity and access for all? I think that's as good a place for us to land as any.
Yeah, I think, that's so, so hemophilia care is kind of like, climbing a 14er, and the closer you get to the top, the more, the more you encounter, false peaks, ridges, and that last little, climb to the peak is the most difficult and the most fraught. And I think, so this is the future that we're looking at. There is no clear sailing. And I think, the issue of data is going to be critical. You say that AI may be able to predict bleeds, but AI is trained on data, and especially for rare diseases, data is a premium. And if we don't feed AI with the data that will allow its, to help in early diagnosis or however we're going to use it, then AI won't work. And so you're absolutely right. Data collection is a premium. I think every bit of clinical data should be research data. And I think that's particularly important for rare diseases. But so that's what our future is going to be. But it's never been a more difficult time to collect data because, data collection also needs funding, and it's been a very, very difficult time. So, we've never needed data more, but we've never been able to truly mobilize and really collect the universal data that we need. And my hope is that somehow we'll find that way. And I want to get back to something that I meant to mention several times actually during our conversation, and I'm glad you brought it back up. And that is global inequity. Because, one of the major risks of innovation is that the price of the cost of treatment goes up. And the more the cost of treatment goes up, the greater the inequity of care across the globe becomes, right? So the more expensive, the less likely individuals in lower and middle income countries are going to be to receive the higher standard of care, the more expensive standard of care. And this is a shame. And because the disparities are only getting greater. Now, I want to say that I have to congratulate the World Federation of Hemophilia on its recent work in getting a lot of our more novel therapies to be in consideration or on the WHO Essential Medicines list. And I think that was great work. And that's a first step in making these therapeutics available to the rest of the world. But we've got a long way to go. And I think we've got to figure out a way with our industry partners, a way to ensure that these therapies are not so expensive that they're so out of reach for so many. I mean, there's no way around that.
So the work continues. Well, from crisis to community counts, from registries to real-world data, from paper logs. Remember paper logs? To digital twins. The last 40 years have taught us that every number tells a story, and every story adds to the data. I've grown up with hemophilia, and hemophilia has grown up with me. The next 40 years won't look the same, but they'll depend on the same truth. We measure what matters because people matter. That will do it here on this episode of the Global Hemophilia Report. I'd like to thank Dr. DiMichele for an extraordinary season this year in 2025, and thank you to the dozens and dozens of contributors we have had on this show. Thank you to Japneet Dhaliwal, Kay Vermeil, Alex Watson, Jade Everidge, and everyone behind the scenes who helps make the Global Hemophilia Report possible. And of course, thank you to our featured advertiser, Sanofi, without whom the Global Hemophilia Report would not be possible. While this episode concludes our 2025 season, be sure you are subscribed so that our 2026 episodes will be delivered to you the moment they go live. You can also revisit all 51 episodes of the Global Hemophilia Report by visiting globalhemophiliareport.com or searching Global Hemophilia Report wherever you get those podcasts. Dr. DiMichele, thank you for everything this season. It's been a pleasure.
And Patrick, happy birthday. You've come a long way from the 9-year-old boy I first met in New York City, and I am so proud of you. And I can't tell you what a pleasure it's been to have worked with you, not only this season, but all the seasons before.
How about that, huh? 51 episodes so far and counting. Well, until next time, thank you all for watching and listening. My name is Patrick James Lynch, and until next time.
Sanofi is committed to bringing new perspectives and bold innovations to the global hemophilia community. Learn more about how Sanofi is committed to breaking barriers and supporting the community at redefininghemophilia.com.